Hi everyone. Today we're going to take a close look at a 2024 study by Liu et colleagues that details a novel molecular mechanism behind age-related hearing loss. Their research points to a specific protein, bone marrow stromal antigen 2, or BS2, as a key driver of the cochlear nerve damage we see in aging and also as a potential therapeutic target.
So we're going to follow the researcher's journey pretty closely. We'll start by looking at the pathology itself, what actually happens to the cochlear nerve as hearing declines with age. Then we'll see how they identified this molecular suspect, BST2, and critically, how they tested if it's actually causing the problem. From there, we'll dive into the downstream mechanism, this cascade involving NF-kappa B and PO6F1, to build a complete picture of the pathway and what it could mean for future treatments.
All right, let's start by examining the core pathology. We're going to look at the structural and functional deficits that appear in the cochlea during aging, with a specific focus on the myelin sheath that insulates the spiral ganglion neurons. What we're looking at here is that primary pathology, the loss of myelin. This slide shows immunofluorescence data from the cochlea of mice at different ages. The green stain is myelin basic protein, or MBP, a core component of that myelin sheath. The red shows the neurons themselves. If you just track the images from the top at 3 months down to the bottom at 18 months, you can see a very clear progressive loss of that green signal. This indicates the myelin sheaths are degrading over time, and the graph quantifies this, showing a particularly sharp drop between 6 and 12 months of age. Now, to get an even closer look at this, the researchers used transmission electron microscopy. On the left is a nerve cross-section from a young 2-month-old mouse. You can see those thick, dark, well-organized rings around the axons. That's healthy, dense myelin. Now contrast that with the image on the right from a 12-month-old mouse. You can see there's significant pathology. The myelin sheath looks looser, the layered structure is starting to disintegrate, and it's clearly thinner. This provides confirmation of myelin decay at the ultrastructural level.
So we've seen the structural damage, but does this actually translate to hearing loss? Well, yes. This is measured using auditory brainstem response, or ABR. On this graph, the y-axis is the hearing threshold in decibels, so a higher value means worse hearing. The blue line from the young 2-month-old mice shows a low threshold, but the red line from the 12-month-old mice is significantly higher across all sound frequencies. This means it takes a much louder sound to get a neural response in the older mice, which confirms a significant functional hearing deficit.
At the same time, something else is happening at the cellular level. The study found a molecular shift in the Schwann cells, which are the cells responsible for myelination. In young mice, these cells express high levels of a protein called N-coherin, which is linked to cell motility. But in the aged mice, the cells switch to expressing high levels of E-coherin, which is associated with a more static, less mobile state. This suggests the Schwann cell's intrinsic ability to repair and maintain myelin is declining with age.
So this leads to the next logical question. What's driving this functional decline in the Schwann cells? To figure that out, the researchers used single-cell RNA sequencing to compare gene expression in cochlear tissues from young and aged mice.
The result of that sequencing is visualized in this TSNE plot. You can think of it as a map of all the different cell types, where each colored cluster is a distinct population grouped by its gene expression profile. The analysis successfully identified 14 different cell populations. This was a critical step because it allowed the researchers to digitally isolate the Schwann cells and analyze their gene expression changes specifically without noise from other cells. From there, they honed in on that Schwann cell cluster. By analyzing which genes were changing with age and then cross-referencing their findings with a public data set to ensure consistency, they were able to narrow down the list of suspects to just 11 candidate genes. Now, of those candidates, they performed a validation using QRT-PCR to confirm the changes. And as this bar chart shows, one gene really stood out. Bone marrow stromal antigen 2, or BST2, was the most significantly upregulated gene in the cochlea of the 12-month-old mice.
Okay, so the gene is upregulated, but what about the protein itself? This image confirms it. The BS2 protein shown here in green is substantially increased in the aged cochlea. And critically, you can see it co-localizes with the red signal, which is our Schwann cell marker, MBP. This confirms the BS2 protein is indeed elevated within these specific cells. And what's more, the plot shows a strong positive correlation between the amount of BS2 protein and the auditory threshold, directly linking this molecular change to the severity of hearing loss.
All right, we have a strong correlation, but correlation isn't causation. The critical next step was to test if BST2 is actually causing the hearing loss. To do this, they needed to specifically reduce BST2 levels inside the cochlear Schwann cells of living animals.
The tool they used for this is an adeno-associated virus, or AAV. It was engineered to deliver a short hairpin RNA, which is a small piece of genetic material that effectively silences the BST2 gene. And to make sure this only happened in the right cells, they put the shRNA under the control of the myelin basic protein promoter, which ensures it's only expressed in myelinating Schwann cells.
And the results of this intervention were quite clear. On the left, you see the ABR thresholds for the control mice, showing the expected hearing decline for their age. But on the right, the mice that received the BST2 knockdown vector show a significant improvement. Their hearing thresholds are much lower, much closer to what you'd see in a young mouse. This demonstrates that reducing BST2 expression is sufficient to rescue age-related hearing decline.
This improvement in hearing was also reflected at the structural level. If you look at the electron microscopy images again, the control mice on the left show that characteristic disorganized and thinning myelin. In contrast, the BS2 knockdown mice on the right show a much healthier structure with thicker, more numerous, and more regular myelin layers. This directly connects the functional rescue to anatomical preservation.
Now remember that molecular switch in the Schwann cells from mobile to static? Well, knocking down BS2 reversed that too. It increased N-cadherin expression while decreasing E-cadherin. This suggests that part of the therapeutic effect comes from restoring the natural migratory and reparative capacity of these cells.
To corroborate these in vivo findings, the team also developed an in vitro model. They used doxorubicin to induce a state of cellular senescence in a Schwann cell line. Just as they saw in the aged mice, these senescent cells showed impaired migration and, importantly, a significant increase in BST2 expression, further strengthening the link between aging, BST2, and Schwann cell dysfunction.
So, we've established that BST2 is a key culprit. The next question is, how is it doing this? What's the specific molecular mechanism that links high BS2 levels to myelin degradation?
To find out, they went back to RNA sequencing, this time on their senescent Schwann cells, comparing cells with and without the BS2 knockdown. These volcano plots show the resulting gene expression changes, and the analysis pointed to one gene in particular, PO class 6 homeobox1, or POU6F1. Its expression was significantly upregulated when BS2 was silenced, making it a prime candidate for a downstream target.
Based on what's already known about BST2's role in inflammatory signaling, the authors formed a specific hypothesis. They proposed that in aged cells, elevated BST2 activates the NF-kappa B inflammatory pathway, and that this pathway then acts to suppress the expression of POU6F1.
To test this idea, they used specific chemical inhibitors. This Western blot shows the protein levels. When they treated the senescent cells with an inhibitor for the NF-kappa B pathway, you can see a marked increase in the amount of PO6F1 protein. An inhibitor for a different pathway, AKT, had no effect. This provides strong evidence that BST2 suppresses PO6F1 specifically through NF-kappa B signaling.
So now we can lay out a clear intracellular signaling cascade. As Schwann cells age, BST2 expression goes up. This increase in BST2 activates the NF-kappa B pathway. Activated NF-kappa B then suppresses the expression of the transcription factor PO6F1, and this reduction in PO6F1 is what ultimately impairs Schwann cell function.
Okay, so the cascade leads to reduced POU6F1. The next logical step is to confirm the final part of this pathway. What is POU6F1 actually doing, and how does that affect myelination?
The central question was this: as a transcription factor, does POU6F1 directly control the genes responsible for building the myelin sheath?
To answer that, they used a technique called ChIP-qPCR, which can tell you if a specific protein binds to a specific piece of DNA, and the results showed a direct hit. POU6F1 was found to bind directly to the promoter region of the gene for myelin protein 0, or MPZ. This is a critical finding, because MPZ is the most abundant protein in the peripheral myelin sheath. It confirms POU6F1 is a direct transcriptional regulator for this essential myelin component.
This is a very elegant experiment. If the problem is the POU6F1 levels are too low, what happens if you just add it back? So they used another AAV vector to overexpress POU6F1 in aged mice.
The result was a significant improvement in their hearing thresholds, mirroring the same effect they saw when they knocked down BST2. This strongly suggests the main benefit of reducing BST2 is that it allows POU6F1 levels to rise.
This next experiment really sealed the deal on the pathways hierarchy. Here, they did a dual virus experiment. They knocked down BST2, which we know is beneficial, but at the same time, they also knocked down POU6F1. The result? The protective effects on myelin were completely gone. The TM images show the return of those disorganized pathological myelin structures. This confirms that POU6F1 is the necessary downstream effector for BST2's effects on myelin.
So when you put all of these pieces together, you get a very clear, complete picture of this mechanistic pathway, and that has direct therapeutic implications.
This diagram illustrates the full axis. During aging, BST2 gets upregulated on Schwann cells. This turns on the NF-kappa B pathway inside the cell, which then suppresses the transcription of POU6F1. Lower levels of POU6F1 mean less expression of myelin protein 0 and impaired Schwann cell migration. Together, these effects lead to the demyelination and hearing loss observed in aging.
So, to sum up the major findings, this study identifies the BST2, NF-kappa B, POU6F1 axis as a key driver of demyelination in age-related hearing loss. The BST upregulation serves as a specific marker for this type of cellular dysfunction. The fact that its knockdown rescues hearing confirms a causal link and identifies it as a druggable target. And finally, working out the downstream pathway gives us the precise mechanism of action.
The ultimate implication here is that targeting BST2, either through gene therapy or with a small molecule drug, could represent a viable new strategy to preserve the integrity of cochlear myelin and slow down or perhaps even prevent age-related hearing loss. And this, of course, opens up a much broader question. Could targeting these kinds of glial cell-specific inflammatory pathways become a new frontier for treating other neurodegenerative conditions? Given that BST2 has also been implicated in diseases like multiple sclerosis and Alzheimer's, the insights gained from the auditory system could have significant implications for age-related neurodegeneration across the entire nervous system.